Hi everyone, welcome. I hope you're doing well and staying safe. This lecture covers the second half of Chapter 3. It also covers a page on prions from Chapter 4 since it's relevant to the topic we're going to be discussing today. As it pertains to proteins, the last topic we'll be covering is the importance of protein folding and also some of the potential effects of protein misfolding. Now recall that there are proteins known as molecular chaperones, which assist in the folding of proteins. And there are two families of molecular chaperones, the chaperones and the chaperones. And I know the names are not great. Now, the family of proteins that we simply refer to as chaperones bind to the hydrophobic regions of a nascent polypeptide. So nascent, that word simply means being born. Now recall from the last lecture that most proteins are amphipathic, right? They have both hydrophobic and hydrophilic segments. So the hydrophobic segments will have a tendency to associate with each other as a result of the hydrophobic effect. So these regions will tend to interact with each other to decrease their interaction with water. But if this happens before the entire polypeptide is synthesized, it could result in protein misfolding. So the chaperones help to prevent this by binding to these hydrophobic segments in the nascent polypeptide. So they help proteins to acquire their correct conformation. The other family of chaperones, the chaperones, provide a chamber, as you can see here, that is hydrophilic, but which essentially separates the protein from the crowded environment of the cell. So the chaperones provide an ideal environment for folding. Now, a protein's function is dictated by its structure, so protein folding is obviously important. To illustrate the effects of protein misfolding, let's talk about a few diseases that are associated with protein misfolding. And Alzheimer's disease is one of these diseases. It's estimated that about 1 in 10 people over the age of 65 will develop Alzheimer's disease, and that likelihood increases as we get older. Now, one of the characteristics of Alzheimer's is the formation of fibrils, and these fibrils assemble into insoluble aggregates of proteins. Now, there are two types of insoluble aggregates that are seen in Alzheimer's disease. One is intracellular, and we'll talk about them in a moment, and the other one is extracellular. The extracellular one, which is shown here, are the amyloid beta plaques. The intracellular are known as neurofibrillary tangles. The amyloid beta plaques form from a protein that is known as the amyloid precursor protein, or APP for short. Now, this protein, as you can see here, is a membrane protein. And the amyloid precursor protein is cleaved by enzymes that are known as secretases. And you can see that there are different fragments that can result from that cleavage. Now, some fragments have segments that are 40 to 42 amino acids long. And these are shown here simply as amyloid beta. Now, these segments have a tendency to misfold and then to self-associate into fibrils, which form the amyloid beta plaques. And these plaques are insoluble. You can see the plaques in this image, and remember that these are extracellular. So they work basically by blocking the synapse between neurons. So they prevent or block communication between neurons, and as it turns out, that communication is essential for the survival of the neurons. So the presence of the plaques causes neuronal cell death. They're both genetic and environmental factors that are associated with Alzheimer's disease. For example, mutations in the amyloid precursor protein, as well as in secretases, and even in genes that are involved in cholesterol metabolism, have been associated with an increased risk of Alzheimer's disease, particularly early onset Alzheimer's disease, which develops at around age 40 or 50. But most cases of Alzheimer's disease are not familial, they're not inherited cases. So these are sporadic cases, and their causes are not well understood, but some of the causes are being addressed. For example, it's well known that repetitive concussions like those that are experienced by professional football players or boxers are associated with increased risk of Alzheimer's disease. And more recent studies suggest that chronic sleep deprivation also may increase our risk of developing Alzheimer's disease. Now, studies have shown that we all make these amyloid beta fibrils, to some extent at least. and that they're clear when we sleep. So they're removed from our brain through a system that's known as the glymphatic system when we sleep. At the same time, they accumulate when we're awake. So that suggests that the less we sleep, the less time the brain has to clear these fibrils. So keep that in mind. While amyloid beta plaques are located outside of the cell, the neurofibrillary tangles are intracellular. They're inside of the cell. And these are aggregates of a protein that's known as tau. Now, it's thought that the accumulation of the plaques leads to the accumulation of the tangles, but research on this is still ongoing. Now, the tau protein normally stabilizes microtubules, and microtubules in neurons form these long tracks in the axon, and those tracks are used to transport neurotransmitters along the axon, as we'll see later in the semester. Now, the tau proteins are important for the formation of these tracks, but if they become hyperphosphorylated, so if they have a lot of phosphate associated with them, then they form these tangles that you see here, and those tangles are going to prevent the movement of material down the axon. So they're going to interfere with the movement of neurotransmitters, and as a result, they're interfering with cell communication, just like the plaques. So together, the amyloid beta plaques and the neurofibrillary tangles lead to progressive cognitive decline because they cause cell death in the brain. Eventually, they also lead to the loss of basic functions like swallowing and breathing, and this is why this disease is fatal. Besides Alzheimer's disease, there are other types of neurological diseases that are the result of protein misfolding. A special type are the Prion diseases. A prion is simply an infectious, misfolded version of a normal protein. Now, the real interesting thing is that the DNA sequence of the prion protein, which is shown here as PRP asterisk, is the same as the DNA sequence of the normal protein. So the DNA of that codes for that protein is the same as the DNA that codes for this protein. So what that means is that their primary structure is also the same. The sequence of amino acids is the same, since that's dictated by the DNA sequence. Now you can see why this is a special case, because we have the same primary structure but a different tertiary structure. So the prion protein has more beta sheet and it forms fibrils, similar to the fibrils that are seen in Alzheimer's disease. And like those fibrils, these fibrils form plaques, and these plaques affect the brain. Now that's in part because the brain has a limited regeneration capacity, so it's more susceptible or sensitive to the presence of these plaques. Now, prions also have another very special characteristic, and that is, as I mentioned, that they're infectious. They're self-propagating. So in other words, they can cause the normally folded protein to become misfolded. So they catalyze the conversion from the normal protein tertiary structure to the abnormal tertiary structure. Now, someone can become infected with a prion by consuming a tissue that has the prion. And there is a set of closely related diseases like mad cow disease, scrapie in sheep, kuru in humans that are caused by prions. And there is also a hereditary form that's known as CJD or Creutzfeldt-Jakob disease. Now, it's not clear what causes the initial misfolding event, but it's possible that it's related to an age-induced decline in the mechanisms that are present in cells to ensure that proteins fold properly. So a decline in the quality control mechanisms of the cell. And that's supported by the fact that in cows, there are some types of bovine spongiform encephalopathy or mad cow that arise spontaneously in older cows. Another interesting thing about prions is that they cannot be destroyed by cooking or boiling. And this is why people are encouraged to avoid eating roadkill, because there's really no way of knowing where that meat came from.